What is the Rockefeller Foundation, and how is it relevant to my essay? The Rockefeller Foundation is a private foundation based at New York City. It was established by the sixth-generation Rockefeller family. The foundation was started by Standard Oil owner John D. Rockefeller, along with his son John D. Rockefeller Jr., and senior's principal oil and gas business and philanthropic advisor Frederick Taylor Gates in New York State on May 14th of 1913, when its charter was formally accepted by the New York State Legislature. In the 1930s to the 1950s, the foundation was one of the largest donors of scientific funding in the world. In modern times, it is no longer in the absolute top worldwide, but however still a significant donor of funding. As of 2015, the foundation was ranked as the 39th largest U.S. foundation by total giving. By year-end of 2016, assets were tallied at $4.1 billion, unchanged from 2015, with annual grants of $173 million. According to the OECD, the Rockefeller Foundation provided $107.2 million for development in 2018. Beginning in 1930, the Rockefeller Foundation provided financial support to Kaiser Wilhelm Institute of Anthropology, Human Heredity, and Eugenics, which later inspired and conducted eugenics experiments in the Third Reich. The Rockefeller Foundation funded Nazi racial studies even after it was clear that this research was being used to rationalize the demonizing of Jews and other groups. Up until 1939, the Rockefeller Foundation was funding research used to support Nazi racial science studies at the Kaiser Wilhelm Institute of Anthropology, Human Heredity, and Eugenics. Reports submitted to Rockefeller did not hide what these studies were being used to justify, but Rockefeller continued the funding and refrained from criticizing this research so closely derived from Nazi ideology. The Rockefeller Foundation did not alert the world to the nature of German science and the racist folly that German anthropology promulgated, and Rockefeller funded for years after the passage of the 1935 Nuremberg racial laws. The Rockefeller Foundation, along with the Carnegie Institution, was the primary financier for the Eugenics Records Office until 1939. As one might imagine, the fact that this foundation was so integral to so much of the scientific funding at the time meant that it had a massive influence over the scientific world. Had it not been for the foundation, the pseudoscientific idea of eugenics would have been regarded as unscientific and unethical as phrenology a lot sooner. In other words, much of the racially based pseudoscience during this time would simply have been underfunded and lose traction, ceasing to exist, had it not been for the Rockefeller Foundation. How have other disciplines, such as physics and chemistry and computer science, affected the development of molecular biology? This quote from page 5 of Kay's book, The Molecular Vision of Life, Caltech, the Rockefeller Foundation, and the Rise of the New Biology, provides a good introduction in my opinion. The new biology generally acknowledged only mechanisms of upward causation, ignoring the explanatory role of downward causation. It would borrow methods not only from physics, mathematics, and chemistry, but also from other fields of life science, genetics, embryology, physiology. The new biology aimed to transcend disciplinary boundaries and employ whatever tools the problem at hand demanded. Although the transfer of techniques between fields was certainly not new, the design of a large-scale program based on interdisciplinary research encompassing several disciplines, was unprecedented. All sciences have, especially in recent decades, experienced an intertwining with other sciences to some extent. In some fields of physics, certain principles of chemistry have been adopted to explain the movement of electrons in circuit boards. Or in chemistry, certain physical concepts such as relativity have been adopted to explain the properties of many of the heaviest elements. Why would biology be any different? Biology has seen a more or less revolution from the 1950s, but especially from the 1990s, regarding various tools that stem from previous discoveries in physics and chemistry. Everything from the ability to, quote-unquote, see the structure of DNA directly with X-ray diffraction, to being able to tailor genetic information with the depletion of enzymes. This seeming invasion of various physical and chemical practices and tools has opened up a whole new world of possibilities for biology that Charles Darwin, Carl von Linné, or Gregor Mendel could not have imagined in their wildest dreams. A fundamental problem in biology, the study of life, has been until the later half of the 20th century a fundamental macroscopic limitation, with some microscopic tools being completely incapable of being able to observe anything less than a bacterium. This was due to how traditional microscopes work. They depend on visible light, and are thus unable to see on a smaller scale than 0.2 micrometers. But with, for instance, X-ray based methods, one can today observe structures in the genome or proteins. Experimentation has also experienced an incredible change. In Mendel's time, one had to perform lengthy experiments with breeding to get an idea of how heredity actually works, fundamentally, such as how certain traits are dominant or recessive. Today, however, we can not only breed with smaller organisms such as bacteria that undergo generations of evolution in sometimes only weeks, but also modify their genome directly to observe how genes are expressed or shut down, sometimes within just days. These advances are not only observable in a lab, but every time a person goes to a grocery store, one can witness the literal fruit of the latest era of biology. The majority of food today is genetically modified. What competing metaphysical notions of what life is for something are there in the different directions in molecular biology? Again, I quote Lily E. Case, The Molecular Vision of Life, Caltech, the Rockefeller Foundation, and the Rise of the New Biology, from page 10. Ultimately, the resolution of this tension binges on the currently unfashionable question, what is life? A centuries-old query that, although by no means intellectually bankrupt, has proved too challenging for philosophers and is deemed unproductive by most practitioners of life science. My own point of departure is that there are multiple biological realities within which life is systematically explained. Various research schools in life science, during either different or the same historical periods, have privileged different scientific representations of life, with representations referred to the totality of biological practices that include the choice of biological system, methods, theoretical frameworks, and interpretations of results. Each program has labored under the conviction that its own representational practice grasps the essence of life. In the book, she presents how molecular biology, at least during its youngest years as a discipline, differed sharply from older evolutionary biology, as well as from ecology. Gone was the focus on the environment and context in which an organism finds itself and is forced to survive in. Gone was also how the whole of the being, its ability to act on itself, its species relatives, as well as its environment, affects whether it can carry on its genome. No. Only the genome and everything it directly affects, such as the cell division whose being has its existence to thank, is relevant. They had gone from a more teleological-friendly interpretation of the evolution of beings, a focus on why, to a more mechanically friendly how. The microscopic as a molecular context and as the largest individual cell was now what would bring out the secrets of life, and almost infinite with the potentials for change, and preferably improvements of life. The rest of the creature was seen more and more almost as just a collection of flesh, whose juice of secrets had dried out. But even in molecular biology, there may have been a difference in how the application of the new tools for biology was viewed. Lily E.K. presents how the Rockefeller Foundation's, quote, "science of man" agenda was more opportunistic than curious. The purpose of funding the scientists' research was to see how the goal of eugenics, to achieve a so-called improved species, could most easily be achieved. However, it is not certain that this cynical opportunism was shared by their scientists. It is important to remember that the Rockefeller Foundation during this time, and also to a large extent today, was crucial for many researchers to have their research paid for, as another quote, page 6, demonstrates. As the correspondence files, reports, and diaries of the officers reveal, during the 1930s to 1950s, the Rockefeller projects became densely interwoven with the scientific agenda of those universities that were heavily supported through the Foundation's molecular biology program. The Foundation's network permeated their academic infrastructures. A significant number of Rockefeller trustees held top administrative positions in the universities. The Foundation officers cultivated advisors and contacts in nearly every discipline and had detailed knowledge of the academic traffic. The officers became central to what in effect became an informal peer review system. It was not uncommon for scientists and administrators to consult the Foundation's grapevine regarding academic appointments, reputations, personalities, travel, and potential projects. There was thus not only a direct control, but also an indirect control over the cultural zeitgeist of the research world. How do the different notions of causality respond to different orientations in systems biology? Systems biology is a branch of biology that emerged after, in a Kuhnian term, a paradigm emerged. The paradigm can be said to be due to a seemingly simple and even desirable problem in biology that arose with the findings of genomics, but nonetheless as a damned but genuine obstacle: data. The sheer amount of data that became available with all the new technology and methodology ultimately imported from physics and chemistry, which resulted in genomics, was at first insurmountable. Johann Söderberg writes about Maureen O'Malley and John Dupre, who wanted to understand the creation of a new scientific conception and formation according to what can be described as a previous universal conception of the modus operandi of genes that has fallen into intellectual ruins. The genes were no longer islands in a sea with bridges in between, but now it turned out that some islands were totally isolated, while some were only peninsulas, parts of a larger landmass. The phenotype was no longer directly linked to the genotype. The very definition of information in the sense of biology had to undergo a reformation. He writes, "The authors identify two camps in the field of systems biology: a pragmatic branch that accepts the new consensus but continues to work with the same methods and empirical approaches as they did before, based on upward causal explanations. The other branch consists of systems theory biologists who have been more influenced by the depth of cybernetic theory and philosophical questions in the style of what is a system? How does something differ from its surroundings, etc.? Investigating such philosophical issues will, according to O'Malley and Dupre, be absolutely crucial for systems biology and its continued development. And with that, I'd like to thank you for listening to yet another one of my essays in video form. The next essay will be about the 3 periods of late modern science. If you enjoyed this video, I would appreciate it if you left a like. If you're not subscribed, make sure you're subscribed, and not just subscribed, make sure you also click that bell, otherwise you may not be notified when I drop a new video. Please also follow me on social media, preferably Twitter, because that's where I post regularly. Anyway, once again, thank you. Bye!